My name's Hannah. I worked in the Stern Lab this summer with my mentor Riley, and we worked on enhancing photosynthesis in maize. So maize is pretty inefficient at photosynthesis in the cold, and we think that that's because of a protein called PBDK, which is involved in this C4 photosynthesis process. But miscanthus, which is another plant that uses C4 photosynthesis, is really chilling tolerant. So we wanted to see what the effects of expressing miscanthus PBDK in maize would be. And we did that by making some plants that expressed both maize and miscanthus PBDK, some plants that only express miscanthus, and some plants that only express maize. And then we took its photosynthesis data and protein accumulation data. And one thing we found was that, especially in low light, it's pretty bad at photosynthesis. The complemented mutants are, even though it's expressing that transgene pretty highly. But more interestingly, I think, is that based on the deactivation and phosphorylation data, we think that it might have a better substrate interaction with the endogenous maize PDRP, which is a regulatory protein, than the maize has with its own PDRP, which is pretty cool. So hopefully in the future, we can try to stack transgenes and overexpress this miscanthus PBDK and improve its photosynthesis, and also test these plants in chilling conditions to see if it conferred that over to the maize. So,
fantastic. Yeah. Do you have any hypotheses about why the miscanthus enzymes might have better interaction with the maize native substrate?
I'm honestly not sure about that. I know that there are very slight differences. I believe it's only 3 super relevant allele changes within or between the maize and miscanthus PBDK. So that's definitely something that could be explored, especially with overexpressing that transgene with stacking different transgenic events. I'm not sure what that difference is, because they have nearly identical structures. So I thought that was really interesting and something we didn't expect.
Great. Yeah, there's always something else to question in science. What was your favorite part of this project?
I did not know anything about corn breeding, and it's really complicated and really interesting. So I liked learning a lot about how CRISPR and crossing genotypes works, and how long it takes to make a genotype of corn. But yeah.
Great. Well, thank you very much for sharing.
Thank you.